show its application in the COVID arena. So over to you, Urashi. Take it away, please.
Great, thank you, Ron. I'm just gonna share my screen and confirm that you can see it.
Perfect. We have your presentation mode.
Okay, great. So thank you very much for conducting this webinar and having us here to present. And just a little bit intro about me. I'm Rajoshi Biswas. I'm the Chief Scientific Officer at Cognita Labs. And my background is actually in electrical engineering. I did my PhD from Rice University in Houston, Texas. But there I was mainly focusing on the lung deposition of aerosols for the 7 years that I was there. And during that time, I co-founded the Cognita Labs company with my colleagues who were actually working on force oscillometry. So now currently at Cognita, I'm taking the lead in the work that we do with our clinical collaborators. And today I'm going to be talking about one of the studies we just started for COVID-19 monitoring with the FOT device, the PulmoScan, which you can see on our screen. And and in fact, the study was really rapidly deployed in a couple of months ago, and every day we are all learning more about this puzzling disease. But one thing that is quite clear is that for some patients, we see that they get affected very severely because of the COVID-19 infection. Some of them are hospitalized. Often they develop complications like ARDS overnight, pretty much. But then you have another huge section of the population which is asymptomatic. And we don't exactly know if they have any internal lung damage. We do have some studies that initial studies that they did some CT scans, and it did show some lungs tissue scarring in the form of ground glass opacities. And I think that kind of shows that it's possible that there is some inflammation or fluid or debris in the lungs that could be causing airway stiffness. The idea is that there seems to be some underlying lung damage, whether it's asymptomatic patients or the severely affected ones. And in fact, from what we learned from the early 2000s with the other SARS outbreak, was that many patients who were followed for about 15 years, they saw that the CT scan still showed abnormality even after 15 years post the recovery from the infection. And for many of the patients, they did have significant pulmonary function loss, which also took 15 years to recover. And in some cases, it didn't actually completely recover. So the main point here is that this disease, COVID-19, seems to affect the lungs, but differently for different people. And our clinical collaborators were really interested to know, does lung function monitoring, would that be able to tell us more about how the infection is affecting people? The only problem now is because of the viral nature of this disease that there are challenges in the current mechanisms of lung monitoring. Particularly, there are concerns about the traditional spirometry being an aerosol generating procedure, so there could be a risk of cross-contamination. And then if you think about it, from what we are learning about the disease, is that a lot of patients get very sick, and it might be very difficult for them to perform a quality spirometry test. So in that way, the traditional PFTs do seem to be not scalable or feasible for this particular disease. And even though CT scans are showing a lot of information about the lung tissue scarring, but then again, it's not really scalable. Maybe it could be used for monitoring long-term damage. We do once every few years, but I think so. Particularly for this disease, it may be not logistically possible. So our collaborators thought, we have FOT, the forced oscillation technique. Why not look at that and see if it could tell us something about the infection and any changes to the lungs because of the disease. And as I think we've all been given a nice intro about the oscillometry technique and the parameters that it measures, the airway resistance, reactance, but because of the kind of data that we saw from CT scan, that there's lung tissue scarring, there's possibility that that could indicate some sort of lung stiffness, and FOT could be, there could be a promise for FOT to pick up those changes in the lungs because of COVID-19. So, and there's a bit of prior work also in ARDS and these kinds of lung complications where FOT parameters show correlation with how the disease progresses. And in fact, in the previous talk also, we saw similar to this. In fact, we have also some prior work that showed that sometimes FOT parameters are also used as a guide for setting ventilation parameters for the patients. So, but not in COVID, obviously. These are in the pre-COVID era. So from the data that we have, we do see some backing for our hypothesis that FOT could be used as a measurement for lung impedance in COVID-19. And it's also likely safer because it doesn't obviously involve in any forced expiratory maneuver. It's just simple tidal breathing through the antiviral filter. So that also gives us some advantage in that. And for this study, we used Cognita's PulmoScan device, which is FDA cleared for both home and clinical use. And it's basically the first completely portable, standalone and wireless, literally handheld FOT device. And obviously, as you know, there's no forced breathing. It's very simple to use and very ideally suited for the kind of study that we did, where we have healthcare workers literally going from patient to patient, room to room, doing the FOT test on each patient. So it's meant to be easily deployable in the field to make these oscillometry measurements, and it also allows us to sort of view and analyze the data remotely because it's cloud supported. And so we use the PulmoScan in this current study. To give a brief intro, we are collaborating. Our collaborators are Dr. Chris Landon from the Ventura County Medical Center here in California. And what they've done in California is they're housing a lot of the homeless population in temporary COVID-19 designated shelters. They've converted some of the hotels in the area into COVID shelters. And what they do is the healthcare workers, they make rounds every day to take symptoms and temperature and some other important such physiological measurements. And what we did was we piggybacked on that and added FOT measurement as another one that they could quickly do. And an important thing here to note is that because it's a shelter facility, we only have mild and asymptomatic patients. The more severe ones are obviously, they have to be hospitalized. So we don't really have access to those patients yet. And it was very quickly deployed, so we don't have funding for the study. So the nurses there are really resource strapped. So we had a lot of constraints that we had to make sure that it could be quickly done and with minimum interference to their existing procedure of doing the rounds. So what they do is they have a kit of devices like thermometer and all of that, and just added the PulmoScan to it. And they go room to room every day, take the temperature, symptoms, and then also do the FOT. And we had to make sure that the safety procedures were in place. So we have our antiviral filters that they use, the nose clips, so that patients only breathe through the filter. And the nurses are always in full PPE. And the study procedure was that they would do one test session per day, and the patient we make sure that they don't touch the device, that's sanitized between patients. And what they do is after they do their rounds to take all the data, they bring it back to the control station, and we can view the data remotely. So the idea is to have minimal things that the nurses have to carry, so no computers or tablets or anything like that. Ideally, in the clinic scenario for FOT, we would like the clinicians to look at the real-time breathing to make sure the patients are breathing correctly, and that helps with the quality control of these tests. But because we were very resource strapped and there were a lot of constraints here, we just had the nurse just observe the patient doing the test and make sure that it was done properly, the breathing was slow and steady. So anyway, so we did this test, and currently we have data from about 4 patients who were monitored over the course of about 10 to 15 days. 2 of them recovered, and 2 of them, I think, were still infected. But we also have 1 control patient. So this is a summary of the results that we have. And as you can see, the first 4 are the COVID positive patients. The last one is a control. And just to give a brief summary of what we observed till now is that you can see that the resistances do have trends, even with the reactances. I've reported AX here. For example, with the first patient, he was actually asymptomatic throughout, but we do see significant changes in the resistances over time. They were monitored for 10 days, and high variability in their resistances and reactances. For patient number 2, there was a big spike on day 3, which then recovered by the time they recovered from the disease. And for the other 2 patients, again, high variability. And it was quite elevated numbers for the whole duration. They still have mild symptoms, but they haven't recovered yet. And for the last patient, last subject was a control, so they didn't actually have COVID, but it shows that we have very small changes and pretty much very flat data. There's low variability compared to the ones who were infected. And to give a sense of what the data looks like over time, I have graphs for a couple of patients here. So this is the patient 1 who's asymptomatic. And as you can see that there's some downward trend from day 1 of their testing to day 10 when they recovered. But because they were asymptomatic, we actually didn't have any outward symptoms, although we do measure some changes in their lung impedance, and both in resistance and reactance. And for patient 2, we saw that there was a big spike on day 3, which then recovered by the time they day 15 rolled in, and they recovered from the disease. And that was the same that we saw the spike both in the resistances and the reactances. But one thing to note is that when it came to the control patient, the variability was very low. And it was mostly a flat response throughout the time that they were tested. So I'm just going to go back so that you can compare the variability in the patients who were positive, although asymptomatic. But then the one who was negative had very low variability. So this is still very preliminary data that we have. We're still exploring this pilot study, and we're sort of trying to understand if FOT could be promising. The data certainly seems so, to measure the lung changes because of COVID-19. And what was promising was that even in asymptomatic patients, we did see significant changes in both resistance and reactances, plus their day-to-day variability was high. And it kind of shows the feasibility of these kind of studies where you could have a mass sort of testing done, where a healthcare worker just went around and did the testing from patient to patient with minimal things with them, like just one device. And they came back, and obviously they were able to sync the data. But I think what now for the future, obviously what we need is much larger sample to see if this is something that happens for most COVID patients, and the significance of the data. And obviously we only have mild and asymptomatic patients. What would be interesting is if we could scale the study to hospitals who have proper health outcomes or health events, and we could correlate that with FOT, see if FOT has any power to predict or detect changes, more severe changes in the lungs. And that's, I think, something that we would be looking forward to. So yeah, so I'll be happy to take any questions. Thank you.
Thank you very much. I think everybody's on the edge of their seats. We've been looking to see if this data will be coming out in the context of COVID. The logistics behind that are phenomenal. So really well done. Rather than go on, I have a million questions for you. I'm going to turn the floor over to Andrew Curry. Andrew, if you could come online, introduce yourself, and then take the discussion from there. I would appreciate that. Thank you, Andrew.
Thanks a lot, Ron. My name is Andrew Curry. I'm one of the clinical fellows at U of T right now, working with Chung Wei. Thanks, Rajoshi, for the talk. It's really interesting. And I think one of the points that I was thinking about when thinking about your talk was, obviously there's great interest in using oscillometry to characterize COVID-19 and the various symptomatic states and progression. And I think for a lot of the same reasons that it's quite exciting. For that purpose, it's also potentially interesting for one of the big problems we've been facing around this pandemic, which is the lack of access to conventional PFTs and spirometry in particular. And I think even in places where prevalence is decreasing, I know for one in Canada we have quite rigorous quality, infection control standards now being applied. So even as PFT labs open up, there's still quite significant reductions in capacity. And, we recently wrote a review, Chung Wei and our Airways group here, about looking at some of the alternative options to conventional spirometry during this time. And I think one of the things we were most excited about was oscillometry, not only for the potential benefits in terms of it not being an aerosol generating procedure, but also even beyond COVID, having more interest in some of the utility for oscillometry, where it might even be more sensitive than spirometry for a lot of situations. And that might even be the case in some of the COVID data you're showing. So I was just wondering if you could potentially talk more about the use of these kind of portable devices in a more broad population of patients, like some of the asthma COPD patients. I know you were talking when we were offline about some of the work screening for undiagnosed COPD in India, for example.
Right. Sure. Yeah. So definitely, and in fact, when we were talking with our clinical collaborators, they were interested initially in doing PFTs for COVID-19, but now that we have the alternative with the FOT plus, because of the promise that COVID-19, it seems that it's affecting the lung tissues more, maybe oscillometry is more sensitive to these changes than PFTs. So definitely they are more excited about using this the oscillometry technology. But even with their clinical practice, I think a lot of them are looking into if oscillometry could be a way to measure even for the regular respiratory cases such as asthma, COPD. And like you mentioned, we've done some preliminary clinical studies with our partners. Actually, in India, we have our clinical partners who have a different use case for this kind of device. So there they often have cases where they need to do mass screening. So there are a lot of undiagnosed COPD patients in remote locations like villages, where they literally have a healthcare worker puts devices in their bags like blood pressure monitor and other such portable diagnostic equipment, and then they go around the village and test like 100 patients a day. So for that purpose, we cannot, so in that case, we don't really have a trained technician who's doing these rounds. So we need the test to be easy, simple to be done. And again, the portability of wellness can help. So they went around and screened 1,500 patients for lung abnormalities. And that kind of use case scenario now gets opened up for oscillometry because of the portable nature of the device. And definitely for the regular clinic use case, if because there are concerns with the PFTs, I think it is promising now that oscillometry seems to be more, physicians seem to be more receptive to FOT because and there's tons of data on COPD as well, that it could possibly be more sensitive to small airway changes and therefore be more useful for COPD. So it does open up a lot of possibilities now with the portable and mobile testing, as well as for the regular clinic use case.
Great. Andrew, you might want to comment on some of the work you guys have done in Toronto to also show heightened sensitivity of oscillometry compared to spirometry in the setting of your post-transplant. Before I get to a quick question about do we have any science? It's intuitive that oscillometry would be not as aerosol generated compared to spirometry. It's one thing to say it and believe it, but do we actually have any scientific evidence that would support it? I think that's a big lacune that could easily be filled. So I see that as one of those urgent needs in oscillation mechanics research at this point in time to go forward into the clinical arena, both in COVID and post-COVID eras.
Yeah, absolutely.
Yeah, I do think that's very urgent need to get some data that shows just how much risk there is with oscillometry and the regular PFTs. Think intuitively, like you said, it makes sense, but I think it'd be good to get some research data on that as well. I agree with that.
And I would urge the other industry members present at the seminar to consider it. I think it's in the interest of everybody. The market share in the oscillometry world is potentially 1 billion individuals. So I think cooperation rather than competition is the key to successfully deploying oscillometry in the current practice, and getting a study like that up and running quickly would help. So industry, I hope that you will be able to help, because when we write to peer-reviewed grant organizations, we usually get back, well, what is oscillometry? What does it mean? Reject, and they go on to the next one. But Andrew, over to you.
Yeah, sorry, you just want me to talk a little bit about the post-transplant stuff?
Yeah, just to give people the heightened sensitivity that you guys are seeing.
Yeah, of course. So I mean, so I'm not personally involved in that research at this point, but more Changwei's area. But certainly, I've read over some of it, and I think there is a lot of promise for oscillometry, in particular in identifying episodes of acute cellular rejection post-transplant earlier than we've conventionally been able to with spirometry. And also, you know, there's a whole area of research in CLAD as well, where it's been seen as kind of a interesting outcome versus, again, conventional spirometry, which might be just insensitive to the early changes in the peripheral airways.
Good. I just wanted to highlight that to industry, to tell them that across different disease categories, using different devices and different groups, we're seeing the same sort of signal. And I think that argues to the robustness of the technology. If there are any other parting thoughts from anybody before we go, I think I will end the meeting. We've gone actually 9 minutes over time, so I thank everybody who stayed. I know some of you had to go because of other commitments. I really would like to thank all the speakers for their excellent presentations. I thank the industry, both in terms of devices and therapeutics, for their contributions. Instead of funding the supper, which I guarantee is a lot more fun, we will have a website where all of this information will be archived thanks to their generosity. And look out for messages from me in the coming weeks, both to pick on you as individuals to give presentations and hopefully put together at least 2 more seminars, which will be as interesting and as pertinent as this one was, if I can give myself a compliment. But I think looking at having listened to the talks, I am really pleased that we did not decide to forego the seminar because of the COVID, and despite the challenges I had earlier this morning scrambling to make sure everybody had a link, I think the Zoom platform worked reasonably well. So with that, I'll end the meeting. Thank you everybody for getting up early and attending, and I look forward to doing this again in 2 weeks.
Thank you, Ron.
Okay, goodbye.
Thank you, Ron. Bye-bye.